So I am excited today to introduce my working group partner in advancing D&I and imaging science through BINA. Andreas Nevarez works at the cutting edge of single-cell cancer biology, quantitative imaging, and artificial intelligence technology. His research uses quantitative cell imaging techniques to identify metastatic cell properties, the leading cause of cancer-related deaths. Andreas has been named a Howard Hughes Medical Institute Gilliam Fellow, a National Academy of Sciences Ford Fellow, and an F-31 Fellow. Andreas received the Ray Thomas Edwards Award for his academic achievement and was recently inducted into the prestigious Boucher Honor Society. He received his Bachelor of Science in Biology at Cal State University, Fresno, and his Master of Biomedical Sciences at the University of Texas Southwestern Medical Center. Andreas is known, Andreas is known for his mentorship and has dedicated much of his academic career to supporting historically excluded students in the sciences. Andreas was awarded a mentoring award for his extensive work advancing and supporting scientists at UCSD. He has led 3 SACNAS chapters across the country and works tirelessly to engage his community. On a larger scale, Andreas is the co-chair of the Bioimaging North America Working Group on Diversity, Equity, and Inclusion across our North American continent. This, combined with his time in the lab, inspires him to provide historically excluded students with the opportunity to perform quantitative biomedical research. Andreas has started a new postdoctoral fellow position at MD Anderson. Well, it's a little bit not so new anymore. Sorry about that. At MD Anderson, where he continues his work of quantitative live cell imaging with single cell omics technologies of metastatic patient biopsies. Welcome, Andreas.
Hello, thank you, Heather, for the wonderful introduction. Thank you for the organizers for inviting me to speak today. A little joke is I'm one of the organizers. So, so I started my postdoc a few months ago and spent the majority of the time defining the project and writing fellowships. It's crazy fellowship season. So I'm going to talk about a few stories around during my PhD and master's around this idea of surveying the metastatic single cell state, but using microscopy to infer information about the cell system and what defines metastasis. So to start off, let's get familiar with metastasis. So metastasis is the spread of cancer. Now we have this primary tumor. The cells in this primary tumor are experiencing nutrition gradients, oxygen gradients, and they need to be able to degrade the ECM in order to migrate out of the primary tumor and get into vasculature. Whether they recruit the vasculature or not, they have to get into the vasculature. And now when they do so, they're experiencing shear force. They're in suspension now. They're not adherent. They have a rush of nutrients and oxygen. Once they survive this, then they need to be able to colonize and thrive in a distant organ, all having their own distinct properties. It's a pretty robust cell to do this. And oftentimes, not all cells are able to metastasize. In fact, metastasis, pan-cancer, only 1% of cells can metastasize. And for melanoma, which goes into the first story, less than 0.1% of the primary tumors of the cancer cells can actually create metastatic tumors. Yet it's responsible for the majority of cancer-related death. Metastasis is seen as this final frontier of cancer biology, and it goes hand in hand with therapy resistance. Oftentimes in the clinic, patients that present with a metastatic disease will have undergone treatment of some sort. So that's why it's hand in hand together as the last frontier. And just last week, a new company spun out of Fred Hutch, focusing on metastasis and taking a different approach beyond the classic sequencing approach. This is important because while sequencing has provided us a wealth of information over decades in gaining insight into cancer and other diseases, we found that their metastasis is not defined by more mutations, not defined by more rearrangements or duplication events or just CNA overall. It's not defined by doubling time. Some other respiration, oxygen consumption, not defined by that as well. We can't find specific like drivers of metastasis. Now, while we know that there's genes enriched for particular pathways like MMPs that will degrade the ECM and allow the cells to start to migrate into the local area, it's not what is defining the metastatic system to be so resilient. And that's the issue, right? Because if we were able to identify that those nodes through sequencing, we'd be able to tackle those specific pieces of the metastatic system. We haven't been able to, and unfortunately, there is no genetic first pattern among the metastatic tumors. And that presents a big problem, but an area for imaging scientists to get into and take a different look at metastasis and uncover these hidden drivers through a sort of top-down approach. And the critical part is identifying the roots of metastatic ability. In order to do this, we need to identify the metastatic features, sort of have develop these quantitative imaging metastatic assays in vitro and in vivo, and learn about these metastatic properties in order to have targeted therapy for the disease and to either prevent metastasis or to stop the disease once it's reached certain organs, to gain information about why those particular organs are, provide nourishing soil for the metastatic tumors. Instead of what we have now, the classic image of a patient presenting with large-scale metastasis of metastatic melanoma and being treated with vemurafenib. The patient looks great. There's still a few tumors left, but it looks like they're going to go into complete recovery. Unfortunately, this is often the case. There's resistance and even further metastasis, and the patient ultimately passed away. And this is a not uncommon truth of those under, with cancer and with the metastatic stage of the disease. And we want to be able to define these properties and to survey the metastatic single cell landscape using imaging. And the way this connects is that for years, there's a lot of groups, the Bacall group and others have looked into cell appearance and the cell state. And they've defined that you're able to infer and gain insights about the interior signaling properties and the interior cell state just by looking at the cell. And it allows us for it to be a proxy, cell appearance to be a proxy for cell state, and here it's metastatic state. I've adapted this Waddington landscape to express this with the underlying signaling networks and genetic heterogeneity that underpin the cell state. And you can imagine here that these cells will start to navigate, and they're early on may become down these canals and are, let's say, in metastatic cells in the liver and the lung. And here, you see that there's these nodes that are enriched and dominate these areas, these canals here. Now, for this particular hump, something that's not as enriched as a smaller node, this could collapse this whole area. This is something we like to talk about as a hidden driver. It is critical for the system, but it's not necessarily going to be upregulated or enriched. But we need to, through our classic sequencing omics profiling, but it's possible, and which I'll show you in the first story, is we're able to define new avenues to investigate molecularly by identifying these cell properties, these cell appearance properties of metastatic cells. So that brings us to our first story, identifying what are these cell appearance characteristics of metastatic cells, particularly of stage 3 and stage 4 metastatic melanoma. So during my master's and kind of first part of my PhD was working on a team to develop interpretable deep learning, which will allow us to uncover what those cell properties are of metastatic melanoma, and using label-free live cell imaging that allow us to predict a stage 4 over a stage 3 metastatic cell. So commonly what we have is we have our live cell imaging, we look at these cells, they look a little bit different. Sometimes the subtleties and the cell appearance is not as obvious as I made it in this infographic. And then we extract features, whether they're engineered cell features using machine learning approaches, like let's say like cell profiler, you have all these engineered cell features that describe the cell. For us, we took a deep neural approach using an autoencoder, and we gain these features. And what often happens is you have a classification output, a TC and a UMAP, and you see your cells nicely are in these little groups. And that's great. It leads for a great publication. You get a fancy paper, you get a fancy graphic. That's amazing. But unfortunately, you don't have the information about what are those properties? What is it identifying? And that's the meaningful part. That's the part that we need to identify and open up that black box in order for us to understand why these cells are able to be more metastatic than their stage 3 counterpart. So what we're doing here is then going to do some image reconstruction based upon these latent cell features that are derived from these images. And that brings us to our little graphical abstract here. This first story is published, so I'm going to go over the highlights. You can check it out in its entirety. A lot of great work was done and effort was put into this paper over quite a few years. And so we have your raw input image. You're going to then encode the information here about the cell appearance, and you condense it. So it's basically taking, it could be seen as like compressing, but you're taking all this big data and you're compressing it into a space, into like a Excel sheet that is going to take all the meaningful characteristics about that initial image. And then based upon that, you can reconstruct or decode using only the important features that the AI thinks are important to properties about this, and reconstruct an image here, an in silico image. As you see, things that you lose are maybe fine granular details about texture, and you get different details. Let's say you're keeping the cell extension. And we were able to use machine learning based LDA classifier based upon these latent feature space to then discriminate between high and low efficiency. So I'll explain. High efficiency is a stage 4 metastatic melanoma, and a stage 3 is low efficiency. So using these are patient-derived xenografts. The patient samples that were developed for this mouse model, it's a near 100% correlation from the original paper. If the patient had metastasis in the lung and the liver, pretty much majority of the time, you would see that in the mouse. So it's a nice correlation. It's important for when we go back and do our mouse studies in this paper. So we were able to discriminate between stage 3 and stage 4. Great, right? The first part's done. There's differences there. There's differences captured by the AI in this latent space. But what are those features? And that's where we can explore the distributions of each one of these 56 features, these 56 dimensions. And so it happened to be just number 56 was the most different, the most variance between the high and low populations. And we then reconstructed the images based off that. What was really cool is we could push cells from a stage 4 space to a stage 3 and vice versa, and turn the volume up to 11, because oftentimes these are subtle feature changes in cell appearance. So we really needed to exaggerate it and go beyond the limits that you see that are on our distributions, go beyond the limits so that way we as human operators can understand what the AI is interpreting as important information. And what we found was the cellular properties that predict stage 4 were pseudopodia and light scattering, intracellular light scattering. Now, the pseudopodia and cancer and metastasis, there's a lot of literature. It's pretty well studied from a lot of different groups, and it makes sense. Obviously, it needs to move through the environment and probe the environment, anchor on, and pull itself through the ECM. But even other groups have seen that the enhanced branched actin formation is critical for micrometastases. So these are when those few cells get to a new organ, a new site, they need to start going through the cell cycle. And this plays a role. Pseudopodia increases the proliferation of survival signals and allows it to get through cell cycle checkpoints to make sure it survives and thrives in that new location. So I'm going to show a little movie, and this is our in silico projected cells based upon that 56 dimension, that 56 one. So this is only looking at number 56. We're going to be playing it on the histogram. So here is that negative 1.25, and our classifier score is 80%. So we're 80% confident that this is a stage 4 high efficiency metastatic cell, and we're seeing these pseudopodial extensions here, these cellular extensions. And now I'm going to be moving it into a stage 3 space. And so it's going the opposite way on our distribution, and you're going to see that it becomes more bald and round. So going back, playing, you see it's losing the cellular extension. And that's how we were able to identify Pseudopodia as this important cell property for metastasis, the stage 4 metastatic melanoma. The other property was light scattering. This is not as well connected. It's to metastasis. To cancer, there's been a few papers out there that talk about how intracellular light scattering is relevant to cancer development and therapeutic intervention, but not necessarily metastasis. And the literature is not as dense as it is compared to Pseudopodia. So this is something, right, that we're able to investigate. It's a new property that we've seen. There's not much literature into it. This is an area where maybe we can identify a hidden driver, something that is really critical for metastasis. And it makes sense intuitively because changes to light scattering are going to be related to changes in intracellular organelles, phase-separated droplets, stress granules, P-bodies, etc. And those things have critical roles in cells being able to survive stressful environments and having a more robust ability to survive different environments. Now here we have the same thing. We have feature 56. This is our classifier score is 0. We know that this is not a stage 4 high. And then we're going to push this into that space. And as you see here, because now we're off the charts, our limits were -3 and 3 on our histogram, on our distribution. So now we're at the off charts. This is the exaggeration part of it. So that way we can really see its intracellular light scattering. This is phase contrast imaging. We've done a lot of corrections and to make sure that we're not focusing on the phase halo. And this is true intracellular light scattering. And we go into more detail in the paper. So if you have questions about that, refer to the paper. Now, obviously, this is awesome. We were so happy. We have the ability to predict metastatic efficiency just based upon the way the cell looks. We even opened up the black box and we were able to uncover the cell properties. Now, the optimal thing is if we want to get this into a clinic, I mean, this is the whole point why we chose label-free phase live cell imaging, because one, you don't have to do any genetic engineering. You don't have to perturb these cells in any way. You kind of can preserve that cell state. We image them on thick collagen, so that way it experiences similar forces other than like glass or plastic. So it kind of allows us to gain more insight into that and see some cell appearance things that are more relevant in vivo. But we need to make sure that we can, if we get a new sample, we can still predict in our AI. It's not like overfit or something just for our images. So based on the literature, went and found out that there's sort of classical metastatic melanoma cell lines, A375 and MV3. We had A375 to be predicted as a high and MV3 to be predicted as a low, meaning less metastatic ability. We're equipped with our mouse model, and I went from imaging and doing some, in this paper, some of the analysis, and then had to go into the mouse room. So it's a bit of a change for me, but it was a fun project and it allowed me to learn a lot of different things. And taking our prediction, we saw MV3, here's the injected into the flank, and this is the primary tumor, and this is doing bioluminescence imaging, and we see that in the lungs, there is micrometastases. So this is indicated by the color. Now, overall, I could not see large, gross metastatic tumors in these organs. It was more defined by some sparse micrometastases. However, for A375, that was not the case. There were large-scale micrometastases, and there was the gross metastatic tumors that were obvious by eye that really took over these organs throughout our cohorts of animals. Now, it was around this time we were finishing up. I was transferring institutions. I was moving to UCSD to finish my PhD. And there had been a resurgence of interest in telomerase, but not for the idea of immortality for cancer, but an identification that there's these promoter mutations, and they actually are enriched in metastasis and possibly have some functional ability. So I decided to look at this with my cohort of mice and see that the one that was predicted to have a less metastatic ability was wild type, and the one that was predicted to have an increased metastatic ability harbored the telomerase promoter mutation. So I wanted to see if this held up in vivo, small sample size, and to look at different cancers. And so we first identified these cell appearance characteristics, but how do these telomerase promoter mutations differ in metastatic ability in vivo? And so at UCSD, it was more quantitative imaging lab, so we didn't have access to large-scale imaging studies, but we didn't need to. Even though these telomerase promoter mutations increased expression and are correlated to aggressiveness and resistance, and not just development and these unfortunate, therapy resistant and aggressive cancers across tumors and across many, many tumors, we see that in just metastatic cell lines, particularly, so these are only metastatic cell lines, that there is a large portion of the wild type that metastasized. Okay, we saw that. But then the telomerase promoter mutation, there's two main variants seen in the clinic. There's other variants, but they're not seen as often. But the two main variants are what I'm going to refer to as 228 and 250. 228 is dominant, whereas 250 is not as dominant, except for melanoma and esophageal cancer. But for our story, melanoma is more present. Okay, well, that mouse that was predicted to be high efficiency actually harbored the C250T, a telomerase promoter mutation. And that is the thing, is there a difference between these two mutations in ability to metastasize? Why is it's more common in melanoma? And even though often melanoma is like UV-induced cancer, these mutations are UV-induced, but there has to be also something, even, esophageal cancer, there might be an ability, increased function that C250T can give. So we wanted to look at it in vivo. So we mined MetMat, which is a recent paper that allowed for the quantitative potential. They quantify the potential and penetrance of a whole bunch of different cancer cell lines in mice, and they specifically looked at metastatic growth in these 5 tissues. It lacked the TPM information, so we just did a meta-analysis of mining papers and databases to find this information. And we found that C250T has more metastatic penetrance and potential across all organs. Now, because there's a couple caveats, because the C250T and C228T mutations need to either be whole genome sequenced or targeted Sanger sequencing, often these cancer exome studies or panels lack the ability to identify this. Second, because that was the case, not all cell lines have been identified in the study to have TPM information at all. Sample size is a bit low. Because 250T is less often seen in the wild, we have lower numbers here. But beyond all that, it was still a difference. There was still an increased invasiveness and aggressiveness for cell lines that have 250T in vivo. And if you look at the organs individually, you see that 250T has a more potential, metastatic potential for the lung and liver, while having more penetrance, meaning taking over the organ better in the liver, brain, and lung. And so we see that there is a functional difference in association, right? I'm not going to say it's causal. There's an association with the 250T variant and increased ability. What we did not see is that it does not correlate with aneuploidy replication rate or mutational burden, as metastasis doesn't in general, like I said in the beginning. So that launches us into the next portion and the next story, which is to what extent do these telomerase promoter mutants play in the metastatic process. And we'll look at it in vitro and to really dissect the connection between our metastatic phenotypes that we've kind of defined previously and others have defined, and these TPMs. So we needed to engineer a cell system that had a low mutation burden, obviously was not telomerase mutated for immortality, and liked being cultured in a dish in some sort. So we chose 293T, cancer-ish. It has, put it in suspension, has EMT properties, can form cancer in mammary fat pads in order to, to create cancer. So it has this ability. So there's some connection, not completely normal. And we were able to engineer these cells to then look at collective cell migration as a functional phenotype, using our deep learned light scattering and morphological features that we defined in the first paper, and looking at clonal heterogeneity. Clonal heterogeneity is something I haven't talked about, so I'm going to go into that next, even though it's really critical for metastasis. If you have a homogeneous tumor and then you have a selective pressure that kills these cells, all of them are going to die. Obviously, that's bad for the cancer, good for the patient, but cancer doesn't want that to happen, creating heterogeneity along the way. However, when it does experience selective pressure, and this could be entering the vasculature, this could be going to a new organ site, or this could be therapy, most cells will die, as we saw in that patient image, but still some cells survive. And they are founding members of the new tumor, and the heterogeneity is based off of these cells. So one is, if you have a clonal evolution from the primary tumor to the metastatic tumor that enriches for the TPMs in about 80% of metastatic tumors across cancer types, then there is more and more evidence that this could be a functionally relevant mutation, despite it just being in a promoter mutation, and it goes beyond expression. And so looking at this, like I said, we made our cell lines. We had to have them be monoallelic, so that we had to have allele resolution, which was also another reason we chose to use 293Ts. It was easier to engineer at the time when we started the project for us. And because the TPMs only are mutated on one allele, they create an epigenetic, heterogeneous epigenetic landscape across the alleles. Telomerase itself, can be expressed in wild type at low levels that often isn't detected, but it's expressed here in our system. And we had these spectrally distinct reporters that are nuclear localized, and we're seeing that if you just look at the images, there's an increased expression, right, of these 228 and 250 variants, TPMs. So looking at this, you sort of looks like nesting eggs. You have the wild type in blue, the 228 in golden, the 250 in salmon. You see that there's a bit more spread. It's a bit further along the x-axis, which is encoding the telomerase promoter mutant allele expression. That's what we see. We see that 228 has higher expression than wild type, and 250 has higher than both. It's a modest increase, about 1.4 times. But we see that in urethral cancer. It's only a modest increase of 1.4 times, but it's still aggressive. It's still highly metastatic, still deadly, and there's still an enrichment. So it goes beyond just expression, even if it is mild expression. And when we look at the to quantify heterogeneity or stochasticity based on this gene, looking at our isogenic populations of our engineered cell lines, we see that 228 is higher, more variance than wild type, and 250 has the most variance of them all. And it creates the most variation and the most heterogeneity within its line. Now, this is first looking at the just expression, right? We had to look at expression. We wanted to see which one created more clonal heterogeneity. But you can monitor more than just expression. So we chose to do next was to look at the other cell features, to look at texture, shape, spread, in addition to expression, and see if we can discriminate between these three cell lines. Remember, there's only one base pair mutation here. It's the only thing that's different between these. So we're sort of looking at, if you drop a pebble in a lake and you look at the ripples, how does it manifest in these various engineered features? The magnitude of change may be low, but compounding over time or in different environments or in the background of other mutations that affect the cell state, they could be quite large, and there is literature to support that. But we want to focus on what is the effect of just that mutation and to see how it manifests into cell features, microscopy-based features. So we bind the TPMs together. Here, this is a 90%. This is wild type. It's fusion matrix. This is 83%. And we base this on an interpretable random forest model, and we did pretty well. This is great. We can identify TPMs just based upon over 200 features using Cell Profiler, so a whole bunch of measuring object modules. And this is also done in Cell Profiler Analyst. So great tool. I also like it so much because of its reproducibility. I just send someone in the lab or send someone who's inquiring about a paper, just the pipeline. And so I really like it. And so that's just a little plug there. And then when looking at the different bins, so individually 228 and 250, we have a classification score of 87% for wild type. It breaks down a bit, but we're still getting really decent classification of these based upon over 200 features. However, though, that is based on engineered, the fluorescence. When I look at the top features, it is standard deviation. It's related to the variance of expression of the mutated allele and the increased expression that dominates. That explains the majority of the variance. But within the top 10 features, there is some texture differences as well. And this is coming from not the nuclear channel, but the phase channel. And so we wanted to see how is texture affecting it, taking, again, a label-free approach to identifying the differences between these cell populations. Now, texture has been successfully used in cancer classification. They do this with histopathology. It's been done with biomedical imaging, such as MRI, CTs, all these things to identify areas that have cancer. We wanted to apply this to a single cell level, which other groups have done, and it can pretty well identify different cell types. And so this is really stretching to see if we can see differences here. I have this little bit depth map, image depth map here to give an idea of texture. So if you imagine, close your eyes and you're touching this cell, there's ridges, there's valleys, there's things that can correlate to the internal network of the cell. And the Allen Institute did this, probably a few years ago, connecting it to intracellular organelles by just staining the organelles and connecting it to the bright field image. This is something similar, except we're not staining anything. We're not doing anything. We're just trying to infer and to see, is there any difference in this manifesting in cell appearance? If this cell segmentation is based on the nuclear, the signal that is localized to the nucleus. You might be thinking, well, you're kind of looking at nuclear only shape or texture in the phase channel. But the way these 293Ts are, even within a group of 4, and you can look at ATCC's website, the borders start to break down pretty quick. And it can be difficult to identify where is that cell. And so traditionally, you would have to add some sort of membrane marker. We were trying to avoid that. So we took this as an approximation of the cell body. And for most cells, it would give us 75 to 90% of the whole cell body. So this relates to more than just the nuclear area. With machine learning, again, we looked at Herelic intensity invariant Herelic features, the GCLCM stats of those features, as well as local binary patterns, which are inherently intensity invariant, to avoid any biases introduced due to the experimentalist or just variations. And I found that using any one of these features individually, we were able to do better than a coin flip. About 57 to 65%. We were able to tell the difference between these populations just off of phase. So that's not high. It's not a huge accuracy there. It's definitely not clinically appropriate. But we start to see that there are differences manifesting, and there's subtle differences, and you don't necessarily need fancy AI to do so. Obviously, I did look into the fancy AI and developed CNNs and VAEs, variational autoencoders, a lot similar to the first paper, and we are teasing out more information from those latent bases. But I think it would be actually inappropriate to think that the first story to generate images based upon the latent space for these type of images would give too much information for us, because there's low difference there. Now, this was done in 2D cell culture, and we all know the homogeny imposed on our cells in 2D cell culture and plastic. So I decided to look at deep learned these light scattering and morphological features in a different experimental modality, and wanted to use imaging flow cytometry to do so. And it's like a proxy-ish for circulating tumor cells entering the vasculature, because they experience that pressure and they're suspended in solution. So I'm hoping that this natural stress will tease out more differences and allow me to discriminate between the populations and affect the state to see how the TPMs are affecting the state. This is the images. These are the bright field. This is pseudo-colored side scatter. So again, relating to the organelles and nuclear content of the cell. And we see that just on bright field images alone, that's 99.7% using a UNet convolutional neural net to be able to discriminate between these populations. That's pretty good. And then we have 85 and 71, and these off targets. You're more often going to confuse these two. Again, they're only different by one base pair, but obviously there's some sort of manifestation under these stressful conditions that are manifesting in cell appearance. Now, looking at light scatter, we do unfortunately not see that trend. It's pretty noisy and messy. You see that C250T does have some discriminability, but it's just not there yet. We are doing some follow-up experiments and trying to see if you're stressing a little bit more. Will there be changes like with adding some reactive oxygen species in order to tease out these intracellular organelle distribution changes? Okay, so the last and final thing is looking at collective cell migration. Increased telomerase expression does have connections to invasion as well as to organelle changes. So there is literature out there. I was surprised maybe by, like many in the audience, that telomerase has a lot of non-canonical roles beyond just immortality for cancer cells. And the idea is that the cells for collective cell migration doing this wound healing assay is that they adapt a migratory behavior. They have to reach new locations. and is very well connected to the mesenchymal, epithelial to mesenchymal transition. And so you have cells, these leader cells sensing something, and they all start to move. And we find that TPMs increase healing rates overall. 228 has more than wild type, 250 has more than both. And we see that this healing rate is sustained over 24 hours, which is how long it takes for the wound to close. Single cell motility metric shows some differences. The magnitude is lower, and you really have to focus on that. So based on our initial machine learning technique, it wasn't one of the high features, but if we look at the lineage tracking using these wonderful tools out there like TrackMate, we're able to then identify different lineage-based differences on motility and how they're passed on. And so there's different dynamics for C228T and 250 versus wild type there. If you look at overall distance just in the wound healing, you see the same thing. 228 has more than wild type and 250 over both, and it's again sustained over time. Now, what's different and what we wanted to do differently was to look at not just overall rate distance, but to see if there's some sort of directionality or speed based on area and space in the actual monolayer. So if we have the edge here, and then we are taking a slice across, that's going to give us distance from the edge. And as we move in, we're getting further into the monolayer. This was done in conjunction. I worked with a student in Asaf Saritsky's lab at Ben Gurion, and we worked together to get some of Asaf's old code going and adapt it for a different experimental system. And we were able to then look and get spatial information. Here's the edge at top relating to this white line. And as you go down in this chymograph, you're going into the monolayer. And as you go from left to right, you're going through time. And 1 through 8 is really encoding time, and then 9 through 12 is encoding spatial variation. It can kind of be seen as these cells start to move, how long does it take for that initial force and information to be relayed to the back of the monolayer. And we see that overall, these are aggregate, but in the preprint that's coming out, you'll see very, very soon, you'll see that we do break this down by the 12 features, but on aggregate, 228 has more than wild type, 250 more than both for speed and directionality in these, because you're able to get coordinated movement of this as well. So there is coordinated movement differences for 250 over the other two. So just to wrap up here, we found that interpretable deep learning, using interpretable deep learning, we can then gain insights on cell appearance and cell state, and have areas to work top down into new signaling areas such as intracellular light scattering. And based on our mouse models, I was able to go forward after the story was published and start working on telomerase promoter mutations at UCSD. And our meta-analysis uncovered that there are in vivo potential and penetrance differences between our TPMs, and that there is, the stochastic single allele imaging highlighted the increased clonal heterogeneity of the TPMs. And again, bringing this back to the label-free features, we found that TPMs do manifest a difference in the system through cell appearance that we were able to identify using these label-free face-based features, as well as spatial temporal collective cell migration differences there. Now, in the current work, all these things were done in vitro on cell lines. Now, here at MD Anderson, I'm able to bring all that experience and bring it to not even biomedical, but I'm surprised even when I got here, there's tumors coming in next to my bench. So it's very new for me. But these clinical samples of metastatic and primary and even non-cancer samples allow us to look at on the single cell level what the cells look like, what they're doing, these appearance-based phenotypes, but even in the actual environment, using spatial technologies to connect the cell appearance and omics. And this allows me to now develop modalities that will allow for true single cell connection of these metastatically important cell properties and to identify hidden drivers of the cell system using a phenotype or morphology-first sequencing approach to then, kind of go through the noise that's typically done from the single cell omics profile. And instead of trying to infer function computationally from the sequencing, we have function first, and then we get the sequencing after. So I want to thank my funding, the HAL Lab, my collaborators at Ben Gurion and at UT Southwestern, and I will take any questions.
Wonderful. Thank you, Andreas. This is excellent. So you have a couple of questions in the chat.
When doing this lifestyle imaging,
From
typical
Miranda.
number of input cell images. Yes. So for the first one paper, we had millions. And for the second story, it was hundreds of thousands. For the first one, it was a bit different because we needed to, we had a lot more samples and cell lines there. And then for the second story, we needed to control for variability day to day, well to well, and which kind of limited, and we needed to all be there because what I was seeing is that the AI was picking up on differences due to that. I had to be really careful with the density, with everything, because there's such little differences. Any variation I could see could possibly be because I was not being precise in my culturing methods throughout. So yeah, there was less, but it was still on the order of hundreds of thousands in order to make any claims. To screen drugs, yes, yeah. So the idea is to create a metastatic quantitative bioimaging assay. So it's quicker, it's more large scale. You can use this. You don't have to sequence anything. You don't have to engineer anything. The idea is to just look at the cell and infer something. So the idea would be to, who has a TPM? All right, well, I just put this drug on. Is it affecting cells with TPMs or not? And that would be a way to then use this as a drug screen. And there's other tools out there that you, like analytical tools, that you wouldn't have to reinvent the wheel, and to be able to analyze that data as well. Resolution, yes, that affect texture. So we were, I was imaging at 20x for everything. It was a mixture of resolution and throughput. We were doing 40 at first, but we weren't getting enough. Like, to get millions of cells for these autoencoders, and even for the second story, it's just not possible at that. I mean, I guess I could be in there all day, but I have kids, so it was like, do I raise my kids or do I do imaging? So that was a bit of a a thing that you had to think about. So it was 20x, and we went from 40x, 20x, and 10x, and then we also convolved using Gaussian blur to see if that was affecting texture, and it wasn't.
It's funny. The new trade-off is now a square with raising kids.
Yeah, I know.
Oh, that's awesome. Oh, you have more questions? Yeah. Oh, got it. Does anybody have any questions? Want to turn on the camera and raise their hands? And this is a link to the paper, right, Vanessa?
I believe so.
Yep,
did I get that
is
correct, Andres?
Yeah.
Awesome. Yeah, there's the link to the paper, everyone.
Sweet. Right. So any closing remarks? I want to say something, but go ahead.
Yeah, I think one of the great things about being a BINA member is I've been able to connect and have these networking opportunities. But also, I think it was during the pandemic, I had just emailed Feather Answered. We started talking about getting involved, and the new things that were happening in BINA. I was able to present at AIM virtually during that year and AIM this year as well. So just being able to have opportunities, because in the labs that I've been in, they're quantitative imaging, but I've always kind of been the lone person that's doing something weird. And so it's nice to have that community and to get some feedback. Even the bioimaging forums that I didn't even know about until I was in BINA, and working with TrackMate and getting some of the features that I want in there directly just from like tweeting with him. Like, this is crazy. And so all of that happened because I just got more involved in my community. And so it's one of the big benefits to just being here and being a member and you get to hear cool stuff now every month from a lot of different groups.
That's it. That is a great segue into something that I want to share. In the working group, with the D&I working group, one of our goals is going to be to bring more microscopy to undergraduates at community colleges and just at college 4 years that are underserved in this area. And so if you want to work on that with us, please join us. We're currently meeting monthly, but we're going to start breaking off into really honing in on that one goal. So with that, we are wrapping up our first speaker in this series. And I think Vanessa put the information in the chat on how to get involved with the working group. So we see some working group members here. Thank you for joining us.